So for this tutorial, I'll start with discussing MCQ, and then after that, I will go to discuss the immobilized enzyme investigation number 3, and then followed by this DNA model question, and then followed by any other questions that you have. So therefore, we will start with MCQ. So Chloe, can you let me know, are there any questions that you want to clarify? Okay, so question 8. So let's look at question 8. Oh, this is a very interesting question. So the graph represents the changes in quantity of DNA present in one nucleus. So this is the keyword. Total number of DNA in the nucleus at different stages of life cycle. Which stage takes place at X? Okay, so this one is very interesting. It starts from egg, right? And then fertilization, where the egg and sperm fuse, then we get the zygote, and then you can see that the DNA content becomes double during the S phase of interphase, because the cell already becomes a zygote, and then it undergoes the cell cycle. So S phase of interphase, so DNA double. Then during which phase that the nucleus, right, will get only half the number is when the nucleus starts to be formed inside the cell. So the answer should be D, telophase. That means you got a cell, cannot draw the cell like that, animal cell, you got a cell, and then there are 2 nuclei in the cell. Okay, so each nucleus will have only will take 1 copy of each. So this is in each nucleus, only take 1 copy. So answer is D, correct? So question 16 is a very good question. The diagram shows the chromosome of a cell at late phase of prophase of mitosis. So during late phase of prophase, all the chromosomes already condensed. So you can see the total number of chromosomes, 1, 2, 3, 4, 5, 6. Altogether got 6 chromosomes. Okay, 6 chromosomes. So the question asks the student, what will be the appearance of the products of this cell division as they enter the prophase of their next division? So if they enter prophase of the next division, it will look the same, isn't it? The prophase of their next division. What, this is mitosis, isn't it? So it should look the same as this one, because they already completed 1 cell cycle and they are in the another cell cycle. So look for 2 cells where the content inside the 2 cells are the same as this one, because it's the next cycle already. So the daughter cell will have same number and same type chromosome as parent cell after mitosis. So therefore, it looks like this 1, 2, 3, 4, 5, 6. It looks like this one contains these 2 cells similar to this cell during prophase. Okay, so for question 16, answer is A lah. Right? Okay. Right. Any more question? So this question 22 is about meiosis, because it's talking about reduction division. So reduction division is actually meiosis, but it's in the A2 syllabus. Okay. So you may not be able to answer this question unless you have learned this in your secondary school. Okay, so you know that in order to form gametes, usually the diploid organism, when they form the gametes, there's a reduction division, means the total number of chromosomes in the cell will become half. Okay, so this process is called reduction division, so which is also called meiosis, because the total number of chromosomes becomes halved when they produce gametes by meiosis. So the question asks the student, where will reduction division occur in the life cycle? So how to see? Very easy. Any cell that's diploid becomes haploid, or meiosis or gamete formation is reduction division. So therefore, from a diploid becomes gamete, usually this is reduction division. So this is correct. Okay. And then from haploid becomes diploid. See, from haploid becomes diploid, right? So hang on. Sorry, from diploid becomes haploid. So this is also reduction division. So answer is S and V. So answer is B. So 22 is B. Okay, let's see whether it's correct. Yeah, 22 is B. Okay. So next question. So now we're going to look at designing experiment to investigate enzyme activity. So this is the third investigation that I've given in the PowerPoint slide for alginate beads, means immobilized enzyme. So the question asks the student to outline. Outline means you just list step by step very briefly, yeah? Outlines. List step by step in sequence very briefly. How you would determine the optimum pH of the enzyme lactase when it is free in solution and also when it is immobilized in alginate bead. So we want to find out what is the optimum pH. So before we do that, we need to know that. So how to provide or how to prepare enzyme solution of different pH. So this is what we need to know. Okay, because we want to prepare solution of different pH. And can you tell me, so since this is a design experiment question, right, the first thing we need to think of is always the independent variable. So when you design experiment, you need to think about independent variable, dependent variable, and also number 3, standardized variable. So think about this, talk about them, you will get the points. So Chloe, can you tell me what is the independent variable in this question? So there are 2 independent variables in this investigation. So you get the first one, you say that it is the enzyme, because you can see that in this investigation, the enzyme tested in 2 forms. Either number 1, let's say A, free in solution, and number 2 is the immobilized. Okay, so this is also considered as independent variable. Now there's another independent variable that's very obvious in this question. How would you determine the optimum pH? That means to say that in order to determine optimum pH, you must test a few pH, right? For each pH, you need to find out what is the rate of reaction, right? So that means you have to prepare solution of different pH. So every time when you deliberately change a variable, so that variable becomes the independent variable. So is the pH the independent variable, Chloe? Is the pH the independent variable? Yes, so pH is an independent variable. So in this case, how many different pH will you prepare if this is an independent variable? What is the minimum that I've mentioned before that you need to prepare for independent variable? Minimum number must be 5, correct? So the minimum number must be 5. So can you tell me what are the pH that you will prepare for this experiment? If it's 5, what are the pH will you prepare? So what are the pH I'll prepare? So I know that pH neutral is 7, acidic is below 7, basics above 7. So the best thing for me to do is to prepare a pH which will range that spread from acid to neutral to basic, right? Because if I do not know what is optimum pH. So therefore, for the 5 pH that I prepare, the one right at the center is 7. So that means the other 2 that I prepare, maybe I'll prepare 2, 4, 6, 8, 10. Can or not? So pH 2, 4, 6, and 10 will give me a very good spread, right? From very strong acid and strong base. So I'll put 2, 4, 6, 8, 10 lah. Okay? Because if you put 5, 6, 7, 8, 5, 6, 7, 8, 9, the spread is not so big. Okay, so I wouldn't know about 2 and 10. So spread evenly to a broader range is better for us to capture the location of the optimum pH. Okay, so now we know that, okay, you're going to prepare this 5 pH. Now there's a special solution that we need to use to prepare pH, right? Which is called the buffer solution. So now we've got two types of enzyme. Enzyme free in solution and also enzyme that's immobilized, and you must make sure that they are placed in these 5 different pH. So what you need to do is that we need to mix the enzyme with the buffer solution. For the enzyme that's free in solution, you can just mix the enzyme with the buffer solution. But for immobilized enzyme, we must immobilize the enzyme together with the buffer solution, okay, which is a buffer solution, within the alginate beads. Within the alginate beads, we must immobilize the enzyme together with the buffer solution already. Okay, so this is the pH that we prepare. Then dependent variable, we already mentioned how to measure the rate of reaction. So we want to find out so what you need to do is that, so now we've got 5 pH, right? So we're going to put, let's say 5 tubes. Okay, so imagine you got 5, let's say beaker, 100 ml beaker, okay, or 5 specimen tube. Specimen tube is smaller than the beaker. So you put in the substrate. So make sure all the substrate got the same concentration. So we've got lactose as the substrate. So 1, 2, 3, 4, 5. So same concentration, same volume. So this is called standardized substrate concentration. And also standardized volume of substrate. So standardized. Then we add in the enzyme, either in this buffer solution mixed together. They already mixed together. So this enzyme in 2 pH 2, 4, 6, 8, 10. So put this enzyme in pH 2. So this pH, okay, pH. So you've got 2, 4, 6, 8, 10. So put in the enzyme to this is substrate. Okay, so these are substrate. So add the enzyme to the substrate, mix, okay, and then start timing. Because the rate of reaction is equal to rate of change in substrate concentration versus time. Okay. So we want to start time, and we need to fix the time. Okay, you either fix the substrate or fix the time. That means you want to find out, you fix the time and find out how much substrate disappear. Or you fix the substrate and you find out how long does it take for all substrate to disappear. So you need to fix one and measure the other because both are changing at the same time. Okay, so can you follow? So here we want to find out, in this case, we are actually measuring time. Sorry, we are measuring how much product is formed per unit time. Because you've got 5 different pH. So for each of them, we fix the time. Let's say, okay, we allow the reaction to take place for, say, 10 minutes. 10 minutes, 10 minutes, okay? So we allow reaction to take place for 10 minutes. Okay, so after 10 minutes, we will take the solution and do the Benedict's test. Okay, we'll do the Benedict's test and see how much product has been formed. So among this 1, 2, 3, 4, 5 reaction mixture, because you mix enzyme with substrate, so this is called reaction mixture. So within this 5 reaction mixture, After 10 minutes, we are going to take the solution to do a Benedict test. Okay, so that means you're going to take 2 cm cube of the solution of each of these, put in a test tube and boil it in boiling water bath for maximum 5 minutes, and then check the color. Very easy. So among the 5 test tube, the one that shows the color with the greatest amount of reducing sugar will show that the pH, which is the most optimum for the reaction, because optimum means maximum rate. Maximum rate means the most amount of reducing sugar. Okay, so this one is easier to do. So what do we standardize? Substrate concentration, volume of substrate, and also enzyme concentration. Okay, because enzyme also affect the rate. So enzyme concentration, enzyme volume also must be standardized. Everything that affects the rate must be standardized. Temperature also must be standardized, because we change the pH, so temperature must be standardized. So if you want to standardize the temperature, you either put all of them on the table, so the temperature will be room temperature. So if your aircon is 24, so we assume the room temperature will be 24 degrees Celsius. Okay, so that will be 24. But if you want to make the reaction faster, if you know what is the optimum temperature, you want to put all of them into a water bath. Okay, it's a bigger water bath. And then make sure that this water bath is the optimum temperature for this enzyme. If you have found out what is the optimum temperatures, say for example, if optimum temperature is 37 degrees Celsius, you want to run this experiment at 37 degrees Celsius, because then the rate of reaction is faster. So therefore, you can fix the time to a shorter time. Let's say instead of 10 minutes, you can put it 5 minutes. After 5 minutes, you can already do the Benedict's test. That is the best way to conduct experiment, is to use minimum time, minimum amount of apparatus materials, and get the most accurate, reliable result. Okay, so this is what we need to do lah. So every time you're asked to investigate experiment, think about independent variable. There should be 5 of them. Think about what is dependent variables. How do you measure? Like in this case, I want to measure the rate. Which one the rate is faster? So how do we measure? Do we measure the rate of disappearance of substrate or rate of production of products? And then if products, we do Benedict's test. And then how to find out which one got the highest rate? And then what other things we need to standardize? Okay, so I've mentioned about these 3 important things for experiment. Do you have any question? So Vincent want to clarify question 18 for MCQ for mitosis. So let's look at the question. During which process does mitosis occur? Very good question. A, the production of antibodies from B lymphocyte memory cells. Okay. Maybe. Okay, let's look at it. Later on, I'll come back to this. B, the production of mucus from goblet cell do not need mitosis, right? Production of mucus from goblets, or actually you have not learned this one. You learn this one when you come to, so we just assume that the goblet cell produce mucus by secretion, isn't it? Anything that the cell, like mucus, supposed to be secreted out of the cell. So it's by secretion. By secretion means what? From Golgi body, right? Secretory vesicle has to butt off. Golgi body has to butt off to form secretory vesicle, which move towards membrane, fuse with it, and release the material. So therefore, this is not by mitosis. Okay, so this is not by mitosis. Then C, the production of cancerous tissue in alveoli. This one very straightforward, see? The production of cancerous tissue in alveoli. So you know that cancer is due to uncontrolled cell division by mitosis. So definitely the answer should be C already lah, not A lah, because production of antibody is similar to mucus. That means the B cell has to produce, because antibody is protein, right? So the B cells inside the cell, they have to produce protein. So the rough ER will produce the protein. So this is the nucleus and this is a rough ER, right? It's highly branched, right? So let's say I'm drawing the rough ER, okay? That is joined to the, let's say, nuclear membrane, okay? So this nuclear membrane. So this is the rough ER, right? With the ribosome on it. So the ribosome will be synthesizing this protein that move along the space, right? This is called the cisterna, right? In the rough ER. And then it butts off as transport vesicle, and then it goes and join to form the Golgi apparatus, right? So all joined together becomes the stack of flattened membrane bound sacs, right? Golgi apparatus. Then from Golgi apparatus will butt off to form the secretory vesicle. Then it will fuse with the membrane, see? And then release the content. So this content can be mucus, right, secreted by the cell. This content can be antibodies secreted by cell, can be hormone, right, like insulin, glucagon. That's how they are being produced and secreted by the cell. Enzymes in our intestine also secreted by cell in this way. So this called secretion, right? Secrete means the cell produce something and release it out of the cell that is useful for us. So it's called secretion. So secretion is always this way, this method here, okay? So antibodies are useful, mucus is useful, so it's called secretion, the same process. D, the production of plaques in the atherosclerosis. I don't think you know what's D. So therefore, when you do not know what's D, you choose the best answer. Best answer is C, full stop. Okay, you do not need to know what is this and this lah, okay? Doesn't matter, choose the best answer, right? Okay, that's for question 18. Answer correct or not? Right, correct. Then what else? What's the next question? We can discuss. 21. So the diagram shows a mitotic cell cycle. When radioactive nucleotides are supplied to the dividing cell, at which point will they be incorporated into the chromosome? So, because nucleotides are radioactive, so wherever they go, they will make that location of the cell becomes radioactive, and we can trace that radioactivity using X-ray film. X-ray film, because radioactivity will expose the film, and then that part is exposed for chemical reactions, it becomes dark color. So that's how we trace it. So when you say nucleotide, so when the nucleotide enters the cell, and the cell uses the nucleotide to make chromosome. So when is it that the cell will need to make new chromosome? In this, let's say, now this circle is a cell cycle, right? And then they label the gray region as the interphase. So tell me, when is it that the DNA will replicate? So that is when nucleotides will be added into the template, both the template of the DNA to make the new polynucleotide strand, right? So that's when the nucleotides will be assembled together to form the polynucleotide strand by semi-conservative replication. So which stage is it? Interphase or mitosis? Because here only got two phases, see? Interphase and mitosis. So which one? Hello?
My answer is B, but that's wrong.
Your answer is B, is it? B,
Yeah.
B
Yeah.
for Bangkok lah.
Yeah.
Okay. Can you tell me B is located in the gray zone or the white color zone?
White color zone.
White color zone is what?
Mitosis.
So DNA replicate during mitosis?
DNA replicate now.
So DNA replicate during interphase,
During
right?
yeah.
Early, middle, or late stage of interphase?
Is it early?
Late. Yeah. Late. You know why? Because interphase got three phases, right? Can you tell me what are the three phases of interphase?
G1S and G2, right?
Very good. So G1, S, and G2 phase, right? So
Yeah.
G1 phase will occupy a majority of the interphase. G1 phase is when the cell will be actively growing. So you synthesize a lot of cell component actively growing. For example, plant cell will be producing what? New cell wall, secondary thickening. Okay. What else? Protein, lipids, carbohydrates. So they'll be synthesizing all these things, mRNA, okay? So therefore, very active. Then cell division is during S phase. So it's later stage of interphase. The answer is A lah. Okay? Okay. Can So next question is question 23. Question 23 is also very interesting question. So the diagram shows the chromosomes of one cell which has been squashed during mitosis. One cell that has been squashed. So therefore you can see the chromosome clearly. Because we squash the cell, then the chromosome can space out, then we can see clearly. So which stage of mitosis is shown, and what is the haploid chromosome number in this species? So there are two questions here. Which stage of mitosis? Now this is a very interesting arrangement. If you see the chromosomes, they look like they're arranged in circle, right? It's not a random arrangement. There is a certain pattern that's very clear, which is circle. So we learned that, you didn't learn that chromosome arranged in circle, right, during mitosis, but you learned that chromosome arranged at the equator. So let's say this is the pole, this is the equator. Chromosome will arrange this way at the equator, because the sister chromatids are supposed to separate to the poles, right? So that's how they arrange, so that the sister chromatids can separate to the pole. So what happened here is we are looking at the cell from the pole. So let's say you're standing here and this is the earth, right? So the earth, if you stand on top at the North Pole and look at it, you see the, what do you call this? They call it metaphase plate, this one here, okay? So you see it as a circle, the metaphase plate, okay? So therefore it's in a circle. So this must be during metaphase lah. So it's either C or D. It cannot be anaphase. Anaphase, so another thing is, you know that anaphase, the sister chromatids already separated and it must be V-shaped. But these are not V-shaped. The sister chromatids are still together. So definitely it cannot be these two. So there are two ways for you to get to the answer. You phase out the incorrect answer, then this is correct. If you're not sure, but you're quite sure this is wrong. So the next thing to look for is it 5 or 10? So did they mention anything? No, they did not mention anything. So let's look at it. 5 or 10. So this one is during metaphase. Altogether, we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 chromosomes, right? 10 chromosomes. 10 chromosomes means what? 10 chromosomes means 2n equals to 10. Okay, so if it's haploid, haploid means n. So if 2n equals to 10, n must be equals to 5, isn't it?
Yeah.
So is the answer D?
If n equals to 5, the answer is C, but I choose D.
Answer is what?
The correct answer is C, but I choose D.
So the correct answer is C, because 2n is 10. Normal cell is 2n. So if it's n is half of 10 is 5 lah. Okay?
Okay.
Okay, so do you have any more question?